In this lecture, we will try to study the mechanism of nitrogen fixation by rhizobia and blue-green algae. Rhizobia is the term given to symbiotically nitrogen fixing bacteria. So collectively, such symbiotic nitrogen fixing bacteria are called as rhizobia. So how rhizobia do nitrogen fixation at the same time? Blue-green algae or BZA, they also plays very important role in this process of symbiotic nitrogen fixation. So in both these, how the thing of atmospheric nitrogen fixation takes place and nitrogen is converted into the more available form which can be absorbed by the plants, for example ammonia. That we will study in this bit. So we will start with the points what we will study in this lecture. So what is the process of nitrogen fixation we will focus on. What are the methods of nitrogen fixation, that is physical and biological nitrogen fixation, how it takes place. Symbiotically, how nitrogen fixation takes place. Symbiotic nitrogen fixation by rhizobia, how it occurs, and the related points for the process of symbiotic nitrogen fixation. We know nitrogen is abundant in the Earth's atmosphere. Then also it is not available to the plants. About 78% of the nitrogen is there in the air. That we know. Then also the plants are not able to absorb that nitrogen, as there is triple bond in between the nitrogen. Dinitrogen is having triple bond, and it is very difficult to fix this nitrogen, as it requires 225 kilocalories of energy to break the triple bond present in between nitrogen or dinitrogen. So available plants on the surface of Earth are not able to fix the nitrogen because of this thing. Can't break the triple bond present in between this dinitrogen. So to make the availability of the nitrogen in the nitrate or nitrite or ammonia form, how it happens, that we will have to see here. And in soil, this nitrogen, atmospheric nitrogen, is fixed by two methods. One method, first method, is a physical method of nitrogen fixation, and second is a biological method of nitrogen fixation. Physical method, so during rainy season, lightning occur, thundering of clouds occur, and the energy of this lightning and thunder is utilized for the process of fixation of atmospheric nitrogen. And about 10% of atmospheric nitrogen is fixed physically by this physical nitrogen fixation process during rainy season. Now second method, that is biological method of. In this method, nitrogen fixation in soil takes place with the help of different microorganisms. For example, bacteria, blue-green algae, actinomycetes, or even mycorrhizae, they take part in this process. Bacteria in leguminous root nodules fix atmospheric nitrogen into nitrates. So conversion of atmospheric nitrogen into nitrates and making nitrates available to the plant roots to absorb is done by these symbiotic bacteria. And thus this type of fixation of nitrogen is known as biological nitrogen fixation. And this is the only major source of nitrogen availability for agriculture, that is biological nitrogen fixation. As the nitrogen fixation is done symbiotically, the type of nitrogen fixation is called as a symbiotic nitrogen fixation. Now we will see how symbiotic nitrogen fixation or biological nitrogen fixation takes place. So process is of prime importance, no doubt, for the life of plant. So mutually, or the type of mutualism between bacteria and the root nodules of leguminous plants or non-leguminous plants. So symbiotically it is done. So both the partners here, for example, bacteria, like group we have seen rhizobia. So this group and the legume plants or non-legume plants, they mutually or symbiotically fixes nitrogen, and about or more than 60% of elemental nitrogen is fixed through this nitrogen fixing process, that is biological nitrogen fixation. Symbiotic nitrogen fixing bacteria are associated with the legume are about or have 14 genera and 98 species. Symbiotic nitrogen fixation is the most efficient, most effective and highly economical process of adding nitrogen into the soil. This fixed nitrogen is taken up by the plant root system and provided it to the plant. And it becomes a good example of mutual benefit or mutualism for both the partners. So no one is harmed, neither bacteria nor the plant. Bacteria get food from plant and also shelter from plant in root nodules of the host plant, and in return Bacteria give fixed nitrogen in the form of nitrates or ammonia to the host plants, which is utilized for fulfilling the metabolic demands of nitrogen. Nitrogen fixers like rhizobia or blue-green algae are known as diazotrophs. Diazotrophs means a microorganism that is able to grow without external source of nitrogen actually. Microorganism which is able to grow without external source of nitrogen. Or also we can call diazotroph as bacteria and archaea that fix atmospheric nitrogen gas in more usable form like ammonia. So we have seen what is the diazotroph. So how symbiotic nitrogen fixation by rhizobia takes place, that we will see now. So this process occurs in the following manner. So in first step, colonization of host root hairs by rhizobia take place, and as a result of what, curling of root hairs take place, and formation of infection thread inside, that is included in the second step. And in third step, the infection thread gets branched and rhizobia are released in the cells, cortex cells of the root. So how it happens, that we will see here. See, first colonization of root hairs of host plant by rhizobia. So rhizobia are here. Collectively, we are calling rhizobia. Actually, these are the microorganisms concerned with the nitrogen fixation. So this is root hair. So root hair is delicate, and through this part, these rhizobia, they can enter into the root hair. So first, or in initial stage, in the formation of root nodule, which begins with interaction of rhizobia with legume plant roots. There is host specificity of bacteria. So particular plant roots will be infected by particular bacteria. This is host specificity. Different types of bacteria are present surrounding to the rhizosphere of soil of host plant. The roots of young host plant secrete a group of chemical attractants like flavonoids and betaines. In response to these chemical signal, only specific type of rhizobia are attracted towards the root. and migrate towards the root hair to initiate the formation of nodule with the help of nod factor. Nod factors present on the surface of migrated rhizobia and bind to lectin proteins present on the root hair surface of the host. Adhesion of rhizobia to root hair completes the process of colonization of root hairs by rhizobia. Now, second step, curling of root hairs and formation of infection thread, how it occur, that we will see in this figure. This is the curling of root hair. The lectin nod factor interaction induces curling of root hairs around the rhizobia. And at these regions, cell walls of root hair degrade in response to nod factors. Immediately, rhizobia enter into the root hair and their invasion in plasma membrane forms the infection thread. So this is that infection thread. Now, branching of infection thread and release of rhizobia. So see here, this infection thread, it goes in the cortical cells. So infection thread gets branched here. Infection thread full of dividing rhizobia elongates through root hair and later gets branched. It reaches into different cortical cells of root hair. Rhizobia are now released in cortical cells either singly or in group. So like this. They get released over there. These bacteria are enclosed by host-derived membrane called as peribacterial membrane. Now rhizobia stop dividing, lose their cell wall and become nitrogen-fixing bacteroids. Further, infected cortical cells divide rapidly to form nodule. Finally, it get connected with the vascular tissues of the root for nutrient exchange. This is how symbiotic nitrogen fixation by rhizobia takes place that we have seen.